All right, we just have a little bit to wrap up. I promise I will not make this last an hour. We need to talk just a little about the meninges, the cerebral spinal fluid, the cranial nerves, and the plexuses. And this should be kind of touching on some of the same things as the lab video. So let's start with the protective coverings of the brain. So in addition to, the skull and the vertebrae, which certainly physically protect the central nervous system, we also have three very different membranes that protect the brain. We have this outer dura mater, and dura means durable, tough, long lasting. It is kind of a little bit like a reusable plastic grocery bag. It's pretty thick. You could not tear it with your bare hands. You'd have to cut it with a scalpel. But it's the outer kind of tough meningeal layer. We have the middle layer, which is the arachnoid mater. If that word sounds like a spider, you're right on. The arachnoid mater gets its name because it has these really fine wisps that kind of look like spider webs. It's kind of cool, but it's in the middle, so it's in the sandwich. And then we have the inner pia mater, which is just a very, very thin membrane layer that is completely just the surface of the brain. So when you see a brain that's been removed and it's kind of shiny and glistening, that's actually the pia mater that you're seeing. It's just a thin membrane on the surface. We will see that there are three extensions of particularly the dura mater that deep dive into the crevices of the brain, and they follow the fissures of the brain. So between the right and left cerebral hemisphere, we have the longitudinal fissure. There's also a deep penetrating area of the dura mater that goes between those two hemispheres. So it kind of follows the contours. There's a deeper penetration between the right and left cerebellar hemispheres. It's not as deep as the falx cerebri, but it is like a little area where there's like a little dip of dura mater. And the tentorium cerebelli is a piece of dura mater that basically goes down between the cerebrum and the cerebellum. So it just totally contours the brain exactly how it is anatomically. If we were to take a look at that, here's our skull bone. Here's a sinus or a big vein that's going to be collecting blood. So let's kind of ignore those things. Then we have basically the brain tissue here down deep. So you can see the outer gray cerebral cortex and then cerebral white matter here. This is a right and left hemisphere of the brain. So this is going to be the longitudinal fissure. So you can kind of see down that deep fissure, the dura mater, which is gray here. There's actually two layers of the dura mater with the vein on the inside. This gray dura mater goes all the way down to the base of that falx cerebri. So the dura mater actually does split apart in certain places. That's where we have a lot of those venous or vein kind of collections. We will also see that underneath the dura mater that there are these little white kind of fine spider webby like tendrils. That's part of the arachnoid mater. This arachnoid mater between itself and the pia mater is the space where we see circulation of cerebral spinal fluid. So the nice thing about cerebral spinal fluid is that it's completely surrounding the entire brain and spinal cord. That allows it to kind of have a cushion, like a fluid shock absorber. It also kind of keeps it floating so that it's not knocking against any of the bone. But that's where the cerebral spinal fluid is found. There are also some of these little invaginations here called arachnoid granulations or arachnoid villi. This is where the cerebral spinal fluid, once it has been sort of circulated and it's ready to be reabsorbed as we're making new CSF, as we're taking away old CSF, it drains through those granulations into the venous sinus, and then those veins return that fluid to the cardiovascular system. The last layer is the pia mater, and it's just a thin epithelial layer on the surface of the brain. You could not separate that from the brain. It just stays right on top. So we've got the dura, we've got the arachnoid and the pia maters. If you've ever heard the word meningitis is some sort of inflammation or infection of these membranes. And remember, the meninges go around the brain and the spinal cord. So we don't have a separate, boundary between those two things. There's no such thing as spinal meningitis, just meningitis. Meningitis implies that there is a problem with the meninges, perhaps a bacteria or a virus, and it would infect that fluid around the brain and the spinal cord. It is incredibly serious. Bacterial meningitis, FYI, if you've not taken microbiology yet, is one of the very, very few bacterial infections that can lead to 100% mortality if it is not treated. Viral meningitis is very different. It almost never kills you, and we don't treat it as aggressively. We kind of give you painkillers because, of course, it's going to be painful. But bacterial meningitis is incredibly serious. You can imagine, because the brain doesn't have a lot of space, any kind of infection, inflammation, swelling is going to put pressure on the brain and potentially cause harm. Back to the show, though. I promised I wouldn't talk for an hour. We've got cerebrospinal fluid, or CSF. It's pretty clear. It would be what I would consider the liquid portion of blood plasma minus most of the proteins. All of the cells have been filtered out. A lot of the electrolytes have been refined, but it does originate from blood plasma. It's just been highly differentiated. It is formed in specialized blood vessels called choroid plexuses. Choroid plexuses are in the ventricles of the brain, so in the hollow spaces, right, in the hollow fluid-filled cavities, particularly prominent in the third ventricle, which is kind of in the center between the two lobes of the thalamus. It's blood vessels that are covered by ependymal cells, that other type of special glial cell that lines those hollow spaces. They do play a role in the maybe fine-tuning of the CSF, is what I would say. Other things that are interesting about CSF, we will see that ependymal cells have incredibly long microvilli, lots of surface area. That is very helpful so that it's producing and kind of aiding in circulating all of that CSF. We will see that there is a blood CSF barrier. It's different from the blood-brain barrier. So remember, the blood-brain barrier is attributed to the tight junctions of the capillary cells and also astrocytes. In the blood CSF barrier, we do need cells, those ependymal cells, to kind of like be one last little boundary so that the brain can protect itself from anything in the CSF that it doesn't want to come into contact with. And it's kind of your last line of defense against something that may have breached itself into the CSF, such as a pathogen, maybe a bacteria or a virus. That could be very, very serious. But two different barriers to keep in mind. If I was to describe the chemical composition of CSF, I would say less protein. Of course, we filtered it out. Less potassium. For sure, less calcium. Remember, calcium has to be super regulated here because it can leak in if it's in too high of a quantity at the axon terminal and inadvertently cause the spillage of neurotransmitter. What we'll see is more sodium, chloride, and magnesium. So it's slightly different in an electrolyte way, but it just has to be perfect so that it doesn't jinx up the environment for the signaling of the neurons. We know that CSF is constantly produced, and the idea is that we take out as much CSF as we put into circulation. So it's coming from the choroid plexuses. The choroid plexuses are located in the ventricles. There's a little bit of choroid plexus in the lateral ventricles, some in the third. In fact, the majority is found in the third ventricle, and some in the fourth ventricle just in front of the cerebellum. So think of those kind of internal like hollow spaces generating this fluid. It circulates through the ventricles down through the central canal of the spinal cord or out around the fourth ventricle in through the subarachnoid space, kind of nourishing the inner and outer part of the brain before being recycled back into the blood supply via those arachnoid villi. Functions of the cerebral spinal fluid: transportation for sure, it makes sense. Everything is carried through fluid. Protection, and I mean that in a chemical and a mechanical sense. Chemical in that it makes the electrolyte environment ideal for the neurons to fire, but mechanical in that it is a very nice liquid cushion as a shock absorber for any kind of head or spinal trauma that might occur. Your brain is kind of floating in this cushion of fluid, which is really important. Buoyancy is another. Think of that fluid as being pretty salty. So the brain is kind of floating in a salty CSF liquid that basically makes it weightless. And that's important because the brain is nearly 3 pounds. It's surrounded by solid bone. There's sense organs. Your head is heavy, right? Just put your kid's head in your lap and feel how heavy that head is. It makes it very easy to create whiplash if there isn't something making that brain lighter. So it is cool that you perceive your head to be relatively weightless. Your brain is particularly happy floating around in its little soup. That's the other really cool function of cerebral spinal fluid. In terms of the CSF circulation, we're going to see choroid plexus structures in the lateral ventricles. Those are going to be on either side, right? We'll also see underneath the fornix at the top of the thalamus, we have the third ventricle in between the two lobes of the thalamus right here. This is where the majority of the CSF is going to be produced, and just a tad here in the fourth ventricle right in front of the cerebellum. So those blood vessels plus ependymal cells are pushing out the fluid. It's kind of coming from the lateral ventricles. It's getting added to the stuff from the third ventricle, down the cerebral aqueduct into the fourth. There's actually a little hole. You can see it right here. It's called a lateral aperture. There's one on each side. That is one outlet for the CSF to come out and then up around the subarachnoid space. Or it can continue down through the central canal all the way through the spinal cord and then out where it becomes the cauda equina at the very, very inferior portion of the spinal column, and then come back up through the space, the subarachnoid space around the spinal cord until it comes back up, and then it will be drained out through those arachnoid villi. So think of the CSF as being generated from the deep inside places of the brain, the hollow spaces, down and then around, drained at the top of the skull. In terms of the cranial nerves, there are 12. I will say some neuro people do have a cranial nerve 0, but we don't recognize that in anatomy because there isn't functional separation. It's kind of a branch off the olfactory nerve. So we like to start at 1, but if you're going into surgical technology, I do want to just put it out there. It's kind of a nuanced, highly special neurological procedure that would kind of parse out like a little branch of that into its own little cranial nerve 0. But for purposes of this class, we're going to go through the 12 cranial nerves, which are olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial. We've got vestibulocochlear, glossopharyngeal, vagus, which is my personal favorite because it's X. I don't know. I just think X, vagus. It just makes me remember it. Hypoglossal, and then accessory. One of the things that's helpful to remember about the cranial nerve position is they kind of roughly go in order. We're starting with the olfactory, optic, oculomotor. We go around, down, and then up. So it kind of makes like a J or a fishhook shape. And they do kind of go in a order that way. I have them listed for you by their Roman numeral, and these should be Roman and not Arabic. We use I, V. Just get more practice with your Roman numerals. Then I have the names. I have their composition, which is if it's sensory information, sometimes it's pure sensory, like olfaction is one way. It's all smell. And it's actually a really large cranial nerve if you think about it. You can actually detect atmospheric chemicals, millions and millions of different scents. It's astonishing. Our ability to smell is actually pretty amazing, and our abilities are not anywhere near as good as things like dogs or other animals have really acute sense of smell. They can smell even more than we can, which is just kind of astonishing. Optic, vision, oculomotor. Oculomotor means eye motor. So these are like the muscles that control the position of your eye. Trochlear. This is also a little bit of eye movement, proprioception, body position. Trigeminal, this is mixed. It's going to have sensory and motor. This is basically sensation and motor control of your face. Abducens, this is also mixed. Some eye movements, a little bit of sensation there too. Facial, mixed. It's going to be some sensory information from taste, but also some, like it sounds like facial. It's going to be facial motor control. Also, saliva production. The interesting thing that I think about the facial nerve that's kind of interesting to me is that this nerve that goes, it's like your sense of taste, it does kind of go through the inner ear area, and one little area of that nerve will actually kind of surface into the middle ear chamber, where really bad ear infections in that area can actually cause damage to your ability to taste, which I think is really interesting. So like ear infections can actually cause a side effect that you can't taste anything. Oh, it's so cool. Anyway, the vestibulocochlear nerve, this is for auditory. I know that you're going to want to say that this is only sensory because you can't like manipulate the muscles that are inside here. However, trust me, there are actually motor neurons in your inner ear. You're just going to have to trust me. I will show them to you in the next chapter. Glosso means tongue, pharyngeal, throat. This is going to be some taste, swallowing, also some salivation. You have multiple salivary glands in your mouth. The vagus, this is really interesting. I think the vagus nerve is kind of underappreciated. A lot of the vagus nerve goes to the gut. And there's too many functions to even list here. But your gut is huge. And we know that things moving through your gut, you have sensation of that. There's motor control of that. This also can play a role in like alertness. People who have like vagal nerve insufficiency can sometimes have trouble staying awake, for example. Sometimes they might like pass out, things like that. It's just kind of an interesting one. We're going to talk about it in multiple lessons this term. Accessory head and shoulder movements, we're kind of moving down. Hypoglossus, hypo, under, glossus, tongue movements. Remember, I just love the tongue because it's the only muscle that doesn't have an insertion. And it's kind of cool how you can manipulate your tongue for speech and also manipulating food and stuff. Anyway, I do want you to spend some time with these cranial nerves. I do care about their composition, and I also care about their function. One last thing before we wrap up this chapter, and that's the plexuses. The plexuses are like where the nerves kind of get all tangled together. A plexus is just sort of a knot. And there are 4. Sometimes people do like identify a 5th. I'll mention that in a second. The 4 major ones are C1 through C5. Those nerves leaving the spinal cord there are the cervical plexus. We have the C6 through T1. This is the brachial plexus. Brachial plexus implies that it's going to go to the arm, right? Cervical plexus is going to be head and neck. Also, a branch of this is going to service the diaphragm. That's one of the reasons why when you get the hiccups or a spasm of the diaphragm, people will try to like swallow or do things like hold their breath or something to try to get that back to normal. Brachial, still pretty high. I do think I like to think of these plexuses as being really key for injuries. So like when you have a neck injury, and most people who fall and break their neck are gonna, potentially be above the brachial plexus. That means they can't move their arms, right? So they're quadriplegic. Above that really determines whether or not they're going to be able to control their own diaphragm and breathe. That's critical. And we're talking C5-6 here. Then we have no plexuses of the thoracic areas because there are no appendages. There's not very many muscles that require a lot of nerves. So we just have these intercostal nerves that kind of innervate that stuff there. We don't see another plexus until we get into the lumbar area. So the lumbar plexus is going to be the nerves that service the anterior leg. So the quads, the front of the legs. The sacral plexus, which is in the sacral or sacrum part of the spinal column, those are basically the posterior leg nerves. Now, a fifth plexus does deserve mention, and that would be the coccygeal plexus, which is just to the tailbone. And we don't have a tail, but embryologically, there is a little bit of a tail that forms before it is fully differentiated. And there are lots of nerve endings in what we would have as the remnant of the tail, which is just the little tailbone. If you've ever injured your tailbone, like fallen on your butt really hard, you know what I'm talking about. It's incredibly sensitive, and there is just a little bit of a branch for the coccyx itself. So take a look at these plexuses. We're going to look more at them in the lab and the nerves that kind of branch off of them, but these are kind of the big plexuses that I want you to be thinking about.